In this video, we discuss about the biosynthesis of amino acids, pentose phosphate pathway, glyoxylate pathway and oxidative phosphorylation. First pathway which we study, that is pentose phosphate pathway. Actually, pentose phosphate pathway is nothing but it is similar to a glycolysis. In this pathway also NADH and pentose like ribose 5-phosphate act as a precursor for the synthesis of the nucleotides. Okay. So first we discuss about the pentose phosphate pathway. So what is pentose phosphate pathway? From name it is clear. Pentose means 5. Phosphate means phosphate group are present. So in this pathway 5 carbon molecules are present that converted in this pathway into a glyceraldehyde 3-phosphate. Okay. And the 5 molecule carbon that are present is ribulose 5-phosphate in this pathway. So the pentose phosphate pathway also called the phosphogluconate pathway and the hexose monophosphate shunt and the HMP shunt is a metabolic pathway that is parallel to glycolysis. Already I told you pentose phosphate pathway is parallel to glycolysis pathway. In glycolysis, you know that in glycolysis conversion of glucose into a pyruvate, right? Breakdown of glucose into a pyruvate. Similarly, in this pathway, breakdown of ribulose 5-phosphate, that is 5 carbon molecule into glyceraldehyde 3-phosphate. Okay? Into glyceraldehyde 3-phosphate. And during this conversion, there is generation of NADPH. Remember this. In this process, NADPH is generated. But in glycolysis, NADH is generated. In pentose phosphate pathway, NADPH is generated, whereas in glycolysis, NADH is generated. Clearly note down this. So this is the pathway. How the ribulose 5-phosphate is converted into 3 carbon molecule, that is glyceraldehyde 3-phosphate. Okay. This is ribulose 5-phosphate, 5 carbon molecule, that is pentose, right? This is converted into ribose 5-phosphate and xylulose 5-phosphate. Both are isomer, ribulose and xylulose. Both are 5 molecule carbon. Now they are converted into a pseudohaptulose 7-phosphate. 7-phosphate, 5-phosphate means phosphate group are present at the 5 number of carbon. 7-phosphate means phosphate group are present at 7 number of carbon. See here. Suppose numbering is starting from here because hydroxyl group are more superior than the phosphate group. So from here 1, 2, 3, 4, 5, 6, 7 and phosphate group are attached to this 7 carbon, means nomenclature of this molecule is pseudohaptulose 7-phosphate. Okay, clear? So ribulose 5-phosphate converted into a ribulose 5-phosphate and xylulose 5-phosphate. After that, they are converted into pseudohaptulose 7-phosphate and glyceraldehyde 3-phosphate, not glyceraldehyde, glyceraldehyde 3-phosphate. After that, pseudohaptulose 7-phosphate converted into erythrose 4-phosphate and fructose 6-phosphate. What? Fructose 6-phosphate. After that, erythrose 4-phosphate. Erythrose means 4 carbon molecule. What? Pseudohaptulose means 7 carbon molecule and ribulose is a 5 carbon molecule. This erythrose 4 phosphates converted into xylulose 5 phosphate. Right? That is similar to this. And this xylulose 5 phosphate then converted by this pathway. Means glyceraldehyde 3 phosphate, then fructose 6 phosphate, then after glyceraldehyde 3 phosphate. And during this all process, which form of energy is released? NADPH. Okay, remember this, NADPH. Which form of energy is released? NADPH. So remember this, in pentose phosphate pathway, NADPH is released, and in glycolysis, NADH is released. And one another question is asked that which cycle is parallel to the glycolysis? Then answer is pentose phosphate pathway, HMP pathway, or phosphoenol pathway. Remember this. These three are similar to each other. Okay, HMP pathway and pentose phosphate pathway both are same. Next is glyoxylate pathway. First of all, what is glyoxylate pathway? Actually, glyoxylate cycle is anaerobic pathway. These are an anaerobic pathway. Actually, these are similar to TCA cycle. Because remember, in TCA cycle, succinate is formed, right? In this pathway also our aim is to form a succinate. What? Our aim to form a succinate molecule. Right? So, the glyoxylate cycle is a variation of the TCA cycle, and it is an anaerobic pathway that occur in plant, bacteria, fungi and protists. What? Plant, bacteria, fungi and protists. Remember this. Question are asked from here. In which organism glyoxylate pathway cycle will occur? Then answer is plant, bacteria, fungi and protists. Next is the glyoxylate cycle center on the conversion of acetyl coenzyme A to succinate. Already I told the aim of the glyoxylate cycle is to formation of succinate molecule. What? Formation of the succinate molecule for the synthesis of carbohydrate. Glyoxylate cycle allows cell to neutralize simple carbon compound as a carbon source when complex source such as glucose is not available. Understand? It means when glucose is not present in bacteria, plant, fungi, then glyoxylate cycle will takes place, and they neutralize the simple carbon compound as a carbon source. Okay, clear? In which organism glyoxylate pathway takes place, and at what condition it will be takes place, and what are the final products of the glyoxylate cycle. Clear this all? These are the pathway of glyoxylate cycle. What happened here? Acetyl coenzyme, already know that pyruvate is converted into a acetyl coenzyme A, and this acetyl coenzyme A enter into a TCA cycle. Normally I told you, normally this acetyl coenzyme A enter into a TCA cycle, but in this glyoxylate cycle, this acetyl coenzyme A enter into a glyoxylate cycle. Similarly, oxaloacetate bind with acetyl coenzyme A and form a citrate molecule. Okay, form a citrate molecule. Now this citrate converted into isocitrate. After this isocitrate converted into glyoxylate and succinate will be removed from here, and this succinate form as a used as a carbon source. And here then glyoxylate is converted into a malate, and during the conversion of this glyoxylate into malate, here also one molecule of acetyl coenzyme is used. So in this pathway, remember this, in this pathway two molecule of acetyl coenzyme A is used. Two molecule of acetyl coenzyme is used in this pathway. Clear? This pathway is clear? Where it is takes place and what is the final product and in which condition glyoxylate cycle will be takes place. Clear this all? So next is oxidative phosphorylation. What do you mean by, first of all, what do you mean by oxidative phosphorylation? Oxidative phosphorylation is nothing but the energy which are utilized or gained in the form of NADH, FADH. Our body cannot take that NADH and FADH directly. Remember this. Our body not utilize FADH and NADH directly. First, after that, NADH and FADH first converted into a ATP. After that, this ATP is utilized by the body. Clear? Oxidative phosphorylation is the process in which ATP is formed as a result of the transfer of electron from NADH or FADH to O2 by a series of electron carrier. The main purpose of oxidative phosphorylation is the formation of ATP from NADH and FADH2, because our body or microbes cannot utilize directly NADH and FADH2. First of all, NADH and FADH2 converted into ATP. What? Converted into ATP. Then after that, ATP is utilized by our body. This process which takes place in mitochondria. Oxidative phosphorylation, where it takes place? Oxidative phosphorylation takes place in the mitochondria. Mitochondria is a powerhouse of the cell. Means all energy are synthesized in the, all ATP are synthesized in the mitochondria. So it is called as powerhouse of the cell. So this process which takes place in the mitochondria is the major source of ATP in aerobic organism. Clear? Already I told you, mitochondria act as a powerhouse of the cell. So this is the cycle, how the oxidative phosphorylation really takes place, and NADH and FADH is converted into ATP. It is a series of complex. What? It is a series of complex in oxidative phosphorylation. That complex is 1, 2, 3, 4, known as the 1, 2, 3, 4. An electron will be transferred from one complex to second, second to third, third to fourth. And after that, O2 gain this electron and they are converted into a ATP. Okay. Converted into water molecule, and ATP will be generated by this. What is ATP? They are a proton. What? Proton. H plus ion. Okay, clear? So see here, what happened here first. See, this is the whole cycle. Okay. This is the complex 1, complex 2, complex 3, complex 4. Here also complex 1 and 4. Actually what happened? There are 4 complex. Complex 1, complex 2, complex 3 and complex 4. Okay. But series is 1, 3 and 4. And 2, 3 and 4. Not 1, 2, 3, 4. Because NADH go via first complex and FADH2 go via second complex. So for NADH the complex is 1, 3 and 4, and for FADH2 the complex is 2, 3 and 4. Clear? For NADH the complex is 1, 3 and 4, and for FADH complex is 2, 3 and 4. So in this complex, NADH is used, and in this complex FADH is used. Clear? Clear this? So what happened here? See, this is the citric acid cycle will takes place here. In citric acid cycle, remember, in citric acid video cycle we studied that NADH and FADH2 both are released in citric acid cycle, right? When malate are converted into fumarate, one FADH is released, right? And after that one NADH is also released. So NADH and FADH both are released in citric acid cycle. So the NADH that is released is go inside the complex first. After going inside the complex first, the proton will be released in intermembrane space. Where? In the intermembrane space of the mitochondria. Then after electron will pass into another complex via enzyme or carrier, that is COQ. They transfer the electron to complex 3. In this complex also 1H plus will be released. Then after cytochrome C transfer this into a 4 complex electron, and here also 1 proton will be released in intermembrane space. And in this electron are combined by O2 and form a H2O, right? Clear? Similarly here also FADH that is released from the citric acid cycle go inside a second complex of the oxidative phosphorylation, and after that electron is transferred to complex 3rd. In this complex proton will be not released in intermembrane space. Proton will be released from complex 3rd and 4th via FADH2. The proton that are present in this intermembrane space, the concentration of the proton will be increased in intermembrane space of the mitochondria. So this electron will go inside in ATP synthase, ATP synthase are present in this mitochondria. Okay. proton will go via ATP synthase in matrix, and in matrix ADP are present. They utilize this proton and convert it into a ATP. What? Converted into ATP. So this is the process of oxidative phosphorylation. How the NADH and FADH are converted into ATP. Clear? Clear this process? The NADH and FADH that are produced by citric acid cycle. In short, I will summarize this oxidative phosphorylation. The NADH and FADH that are generated via all metabolic pathway. This NADH and FADH are not directly utilized by the body. So first, for energy, the NADH and FADH converted into ATP. So for the conversion of this ATP into a ATP, a process is there, that is oxidative phosphorylation. In oxidative phosphorylation, there are series of complex, that is 1, 2, 3, 4. So 1 is for NADH and 2 is for FADH. So the series for NADH is 1, 3 and 4, and for FADH 2, 3 and 4. During the transfer of electron from one complex to another, proton will be released into an intermembrane space. During the conversion or transfer of electron from one complex to another, the proton will be released in intermembrane space. So the concentration of proton will be increased in intermembrane space. So for maintaining the concentration, the ATP synthase are present. ATP synthase accept this proton from the intermembrane space and pass to matrix, pass that proton into a matrix. A matrix ADP are present that combined with the phosphate and this proton and converted into ATP. So these are the process how the NADH and FADH are converted into a ATP. Clear? This is the oxidative phosphorylation. Now another topic is biosynthesis of amino acid. What are the metabolic relation of amino acid? Actually, our body totally depend on the protein, right? And the protein, how the protein is synthesized? Protein are synthesized from the amino acids, and how this amino acids are synthesized in our body. So we study in this biosynthesis of amino acids, how the amino acids are synthesized in our body. Actually, amino acids are also a two type, essential amino acid and non-essential amino acids, right? So the pathway of essential and non-essential amino acids are different. Pathway of essential and non-essential amino acids are different. So first of all, these are the body protein, right? These are the body protein that contain amino acid, and these are the ingested protein. Ingested protein means the protein we take from outside or during our food. So this protein and this protein contain amino acids, right? And what are this type of amino acids? Purine, pyrimidine, what contain this amino acid? Purine, pyrimidine, porphyrin and thyroxine, right? Purine, because amino acids are made up of nucleotide. Nucleotide means purine and pyrimidine, right? Or they are neurotransmitter, phospholipid and coenzyme. Clear? Now what happened this amino acid? This amino acid release amine group because protein contain NH2 and COOH group, right? So after degradation, the amino acid release NH This NH3 converted into urea. And during the degradation of the protein acetyl coenzyme A will be released. After releasing of acetyl coenzyme A, the fatty acids and other lipids will be released. And this acetyl coenzyme A go where? This acetyl coenzyme go into a TCA cycle, right? And in TCA cycle, during TCA cycle, energy will be released, right? And from amino acids, this glycolysis, TCA cycle or process will be takes place. So all these are correlated with the each other. If one process will be stopped, means whole our metabolic process will be disturbed. Clear? If one process will be stopped from this, means the whole metabolic process will be disturbed. So for proper activity and proper function, all metabolic process will take place at their proper function. So what type of amino acid? Already I told mainly two types of amino acid, essential and non-essential. Essential amino acids means the amino acid which are not synthesized in our body, that amino acid is known as essential amino acid. Because we take that amino acid from our food. That amino acid is histidine, isoleucine, leucine, methionine, phenylalanine, threonine, tryptophan and valine, right? And non-essential amino acid, in non-essential amino acid is the arginine, asparagine, glutamine, glycine, proline, serine, tyrosine, alanine, aspartate, cysteine and glutamate. Means 10 are essential amino acids and 10 are non-essential amino acids. Now overview of the biosynthesis of the amino acids. How the amino acids are synthesized. The carbon skeleton of all 20 amino acids are derived from just 7 metabolic intermediates that together are found in 3 metabolic pathways. Means all amino acids, biosynthesis of all amino acids takes place by a 3 metabolic pathway. And the pathway, that 3 metabolic pathway are 3 glycolytic pathway intermediate. First 3 glycolytic pathway intermediate, that is 3 phosphoglycerate. Remember this all. 3 phosphoglycerate, 3 phosphoenol pyruvate and pyruvate. These are the intermediate of the glycolysis pathway. 3 phosphoenol pyruvate and 3 phosphoglycerate. These are the first metabolic pathway. Next is pentose phosphate pathway. Already I discussed in first slide, that is pentose phosphate pathway. So the intermediate of pentose phosphate pathway that are used is ribose 5-phosphate and erythrose 4-phosphate. What? Ribose 5-phosphate and erythrose 4-phosphate. And next is 2 citrate cycle intermediates. That is alpha ketoglutarate and oxaloacetate. Remember these 3 metabolic pathway. What are the 3 metabolic pathway that help in the biosynthesis of amino acids? First is glyoxylate pathway. In glyoxylate pathway, which intermediate is 3 phosphoenol pyruvate, 3 phosphophenol pyruvate and third is pyruvate. And in pentose pathway, second is pentose phosphate pathway. In pentose phosphate pathway, which molecular intermediate act as intermediate? That is ribose 5-phosphate and erythrose 5-phosphate. Okay. that is ribose 5-phosphate and erythrose 5-phosphate. Now next is 2-citrate. Next pathway is 2-citrate cyclic intermediate. In citrate cycle, the intermediate is alpha ketoglutarate and oxaloacetate. What? Alpha ketoglutarate and oxaloacetate. So remember this pathway. Now first synthesis or biosynthesis of non-essential amino acid. How the biosynthesis of non-essential amino acid will be takes place? The basic step is transmination of alpha keto acids that are available as common intermediate. All except tyrosine. All non-essential amino acid except tyrosine are derived from one of the following common intermediate. Means that is the common intermediate of that 3 pathway means glyoxylate pathway intermediate, pentose phosphate pathway intermediate and TCA pathway intermediate. Right? So, except tyrosine, all are synthesized by 3 intermediate. That is, first is pyruvate, second is oxaloacetate, third is 3-phosphoglycerate and fourth is alpha ketogenic. Okay, clear? And essential amino acid. In essential amino acid, their alpha keto acids are not common intermediate. In this, actually, why this are not synthesized by our body? Because the enzyme that required for the synthesis of this essential amino acid, our body lack that enzyme. That's why our body lack that enzyme. That's why these essential amino acids are not synthesized in our body due to lack of the enzyme. So remember this, non-essential amino acids are synthesized by these 4 intermediates except tyrosine. Except tyrosine, remember this. Except tyrosine. These are the amino acid, how the amino acids are synthesized. These are the oxaloacetate intermediate. This converted into aspartate into asparagine, methionine and threonine, and this threonine converted into isoleucine, okay, and then lysine. Okay, these are the amino acids. Phosphanol pyruvate plus erythrose 4-phosphate, they convert into a phenylalanine, tyrosine, tryptophan. This phenylalanine, then tyrosine. Okay. This pyruvate converted into alanine, valine, leucine. Ribose 5-phosphate converted into histidine. Alpha ketoglutarate converted into glutamate, then glutamate converted into glutamine, proline, arginine. And 3-phosphoglycerate is converted into cysteine and glycine. So remember this intermediate. These 5 intermediate are most important. By which intermediate, which amino acids is synthesized? Remember this all. These are the pentose phosphate pathway. This is the pentose phosphate, this is glycolysis and this is glycogen metabolism. This is pentose phosphate pathway. In this pathway, which intermediate is used? Which intermediate is used in this pathway for the synthesis of amino acids? Erythrose 5-phosphate, right? And in glycolysis, which pathway, which intermediate is used? Pyruvate is used. glycerate is used and phosphoenol pyruvate is used. These are used for the synthesis of amino acid. These are the amino acids. And here citrate is used for the synthesis and alpha glutarate is used, right? Sorry, oxaloacetate is used and here 2-ketoglutarate is used for the synthesis of amino acids. So these are the amino acids which are synthesized by this pathway. Clear? These are also a pathway by which amino acids is synthesized. See here. This is the TCA cycle, right? This is the TCA cycle. TCA oxaloacetate is present. This oxaloacetate converted into aspartate, and after that this amino acids is synthesized by this lysine, leucine, methionine, right? And this from alpha ketoglutarate, glutamate is converted into this, and after this arginine, histidine, glutamine, proline is synthesized. From pyruvate, this is the glycolysis pathway, and from pyruvate alanine is synthesized. And phosphoenol pyruvate, this tryptophan, tyrosine, phenylalanine is synthesized. And from 3-phosphoglycerate, glycine and cysteine is synthesized. And from pyruvate, leucine and valine is synthesized. So these are the pathway of the biosynthesis of the amino acids. Now, biosynthesis of aspartate. Aspartate can be synthesized by the body, so it is classified as the non-essential amino acids. In human, aspartate is the most frequently synthesized through the transamination of the oxaloacetate. Means aspartate is synthesized by the oxaloacetate, right? Means oxaloacetate are important for the synthesis of, these are the oxaloacetate, and oxaloacetate are converted into aspartate. So the intermediate that they utilize in this biosynthesis of aspartate is the oxaloacetate. Now biosynthesis of aromatic amino acid. Aromatic amino acids are phenylalanine, tryptophan, tyrosine and histidine, right? Already we see that the intermediate that are used for the synthesis of this aromatic amino acids is which intermediate takes place? Actually these are synthesized by Korismate pathway, not by that intermediate, right? So these are the pathway by which these amino acids are synthesized. Okay, this pathway is known as Korismate. See, here are 3-dehydroshikimate that is converted into a shikimate, and then this converted in shikimate 3-phosphate, then 5-enol pyruvyl shikimate 3-phosphate, then Korismate. Okay, then Korismate. Actually, these are the intermediate by which this essential aromatic amino acids are synthesized. So Korismate is the converted into phenyl pyruvate. Now Korismate is converted into phenyl pyruvate in 2 steps, and phenylalanine is synthesized by a transamination reaction with glutamate. So Korismate is the intermediate by which aromatic amino acids are synthesized. These are the pathway how the aromatic amino acids are synthesized by transamination. Aminotransferase, right? So this all about the biosynthesis of amino acids, pentose phosphate pathway, and glyoxylate pathway, and the next most important is oxidative phosphorylation. So hope clear this all topic. So thank you.